Hi everyone, welcome to Chapter 1, an Introduction to Biology. In this lecture we are going to do a couple of important things. We're going to, one, talk a little bit about what biology is. Two, probably one of the most important things in this chapter is talk about the scientific method. And then three, we'll just finish up with a few last little tidbits to help you get up to speed here on the topic of biology. So for starters, what is biology? Well, biology, the word literally means the study of living things. Bio means living, ology means the study of. But biology is an extraordinarily diverse discipline. There's all sorts of branches. There's things like, molecular biology, where you use chemical assays to determine the structure of proteins and different molecules. There's things like paleontology, where you're studying the remains of living things, so fossils. There's things like ecology, where you're studying how a variety of different species interact within a community. There's plant biology. There's microbiology, the study of invisible little microscopic things. There's the study of physiology, how the body functions. There's a whole bunch of different facets of biology, and my hope for you is that you'll eventually land in one that is most exciting to you. The process of science, all right? Before we get into talking about how science is done and what exactly it is, I want to just give you a brief history here that kind of sets the stage for why science is important. Many years ago, for a very, very long time, the common way that people believed things was kind of referred to as natural philosophy. What we believed about the natural world was basically what the philosophers told us. Okay, one of the most notable of these philosophers is probably Aristotle, and you can see this is a long time ago, about 2,400 years ago. And in either case, Aristotle came up with a bunch of ideas about how the natural world works, and for thousands of years, people just believed what Aristotle said. Here's an example of some of the things that he put forth. He said that heavier objects would fall faster than smaller objects. So if you went up and dropped two objects, one a bowling ball and one a golf ball, the bowling ball would fall faster. And around 1500 came on the scene this guy, Galileo Galilei, who was just a brilliant thinker, and he wasn't quite sold on Aristotle's belief that heavier objects fall faster than smaller objects. And so he set out to actually try and disprove this. And legend has it that he climbed up top the Leaning Tower of Pisa. Here's Galileo. He dropped two things, one a heavy one and one a smaller one. And he wanted to see when they landed, and found out that they landed, in fact, at the exact same time. That all objects, whether heavy or small, are subject to the same force of gravity, so they fall at the same rate. So this was kind of a really landmark idea, because it was one of the first and most notable times where somebody said, no, I'm not just going to believe what the philosophers have told us, I'm going to put it to an experiment and actually see if it's true or not. And this is all happening during kind of the Renaissance period, which is characterized by this idea that we don't have to listen to what people tell us. We can find out for ourselves what is true based upon what's reasonable and what's logical. So that was a famous experiment. Another one of Aristotle's conclusions was that all of the heavenly bodies, the stars and the moon included, were perfect. And they're perfectly spherical, and they didn't have any blemish. It was thought anything in the heavens was kind of the realm of perfection. And Galileo, again, says no. He has a telescope that he's invented, and he studies the moon and finds out that it, in fact, has a bunch of craters and things in it. There's a bunch of pits and places where it's been damaged and collided with, or comets or whatever. But the point is that he's saying no, it's not a perfect without blemish, sphere without blemish. It actually does, in fact, have some imperfections to it. And then another, just an example to drive the point home, is that Aristotle believed that all colors come from white and black. So the white and black are the really only true colors, and then if you just change them, they they have the appearance of blue, yellow, red, or orange, but in reality, in fact, white and black are the only colors. And Newton comes on the scene and he disputes this idea. He actually says, no, white is a collection of a bunch of individual colors that in their own right are their own colors. So he used prisms to shine light. He'd shine light through prisms and find that they in fact form all of these separate colors which together make up white light. So a couple examples of how in this whole time, kind of the Renaissance period and the Enlightenment that followed, you have this kind of movement of people who are deciding that they want to put ideas to the test and see if they hold up rather than just believing them blindly. And so that's what we kind of refer to as modern science. It's kind of the systematic approach of coming to a belief and determining if something is true or not based upon experimentation. And if you can sum up kind of the mind of a scientist, I always appreciate this quote here by Henry David Thoreau. Rather than love, than money, than fame, give me truth. And scientists are generally people that they don't want to believe what is comfortable. They don't want to believe what's easy. They want to believe what's ultimately true. And so if you find that that's kind of the way that you think about life, that you're well suited for the pursuits of science. In either case, how we learn about things is now known as the scientific method. All right. Now you can't just go out and come up with any old experiment that you want to try and disprove some theory. Coming up with a scientific experiment is actually quite a challenging task. And I'll say even a lot of professional scientists out there who are trying to create experiments, they don't even come up sometimes with the best experiments. So there is kind of a specific method to how we go about this, and this is a really important thing in our studies of biology to kind of grasp, because some of you might go out and do research later on or find yourself making experiments, and it's important to know how to set up a good experiment. So this is a method that was first proposed by this guy, Francis Bacon. You don't have to know kind of who he is or his dates, but I just put it up there for interest. Francis Bacon is the guy that kind of coined that quote. You've probably heard it. I think it goes something like, with great power comes great responsibility. Yeah, that was Francis Bacon. In either case, this is kind of a method that he proposed about how we as scientists should go about making experiments. It always starts with an observation. Okay, you have to observe some sort of natural phenomenon. So a good scientist is actually someone who also is kind of a curious person, somebody that wants to know why things are the way that they are and observes interesting things and wants to know more about them. So just for the sake of kind of giving you some context here, I've created an example. Let's say that I, the instructor of general biology, Bio 1740, realized that for whatever reason, the test scores of my students in my class have recently improved. It's some semester and I'm grading exam one, and all of the exam ones are just like super high scores. My students are doing really well. Well, a natural thing for me to wonder is, why is that the case? So the next step of the scientific method is to ask a question. And so my question would be, well, I'm wondering why my test scores have recently improved. Okay. Now, the next part of the scientific method is to form a hypothesis that answers that question. A hypothesis is basically a fancy word for an educated guess or an possible explanation for why the phenomena is the way that it is. So at this point, this is where the scientist has to do a lot of kind of research, a lot of thinking. So I've asked the question, why are my test scores getting better? So I'm going to sit down and think for a minute about all of the possible reasons why that might be. And let's say as I'm doing some research, this is kind of a silly example here, but as I'm doing research, I realize that there's a new product that a lot of my students are recently been enjoying called 5-Hour Energy. All right. I don't know if you've ever drank this stuff. I haven't, but I've heard it tastes terrible. But in either case, let's say I see a bunch of my students drinking this energy drink. Okay. And perhaps my hypothesis is that the reason my students' test scores are being improved is because they're consuming this drink which is giving them extra study focus, and they're able to cram, long hours the night before the exam, whereas before they couldn't. So every hypothesis is going to have one important feature. It's going to identify two variables, which are known as the independent variable and the dependent variable. Okay, and the dependent variable is the one that depends on the independent variable, if that makes sense. So in this right here, which is my hypothesis, I'm wondering if you can spot which one is the independent variable and which one is the dependent variable. Take just a quick minute to see if you can't figure them out. All right, so the dependent variable is this one right here, the test scores. And the independent variable is the consumption of the energy drink. Okay? And you can see that I'm saying that the test scores of my students depend on whether or not they're consuming this beverage. And if you get these backwards, it won't make sense. I'm not saying that depending on what test score they get, they automatically just involuntarily grab up an energy drink and start, chugging it. They get an A on the test, and then without any choice of their own, their body just starts making them consume 5-hour energy. No, that's not the case. But I am saying I'm thinking that the test scores depend on the consumption of the energy drink. So that makes the dependent variable and the independent variable. These need to be in the hypothesis. And far too often, scientists who are getting involved in really complicated studies, it's easy to lose sight of what the actual hypothesis is and to kind of hone down on what the actual independent and dependent variables are. So you need to have these clearly defined, and then the next step is to create a prediction of how those variables interact with one another. In the hypothesis, I've only said that I think there's a relationship between test scores and consumption of an energy drink, but I haven't stated how I think that they are related. So when you make the prediction, you're actually going to predict the causal relationships between them. So an example would be something like this, that I think my test scores in my class will increase if there is an increase in the consumption of 5-hour energy. So in this relationship, I'm proposing that the variables have a directly proportional relationship, meaning if I increase one, I'll increase the other. That's not always the case. Sometimes you can have an inversely proportional relationship where if I increase one variable, it will decrease the other variable. But in this case, it is directly proportional. I'm thinking if I increase the consumption of 5-hour energy, it will increase the test scores. All right. So now the last, or not the last step, but then one of the biggest steps here is the construction of the experiment. So I've come up with a prediction of what's going to happen based upon my hypothesis that test scores really are affected by 5-hour energy. So now it's time to put it to the test and design an experiment to see if I can confirm my prediction or not. And every experiment worth its weight in salt has to consider a couple of things. First, again, they have to clearly identify the independent variable and the dependent variable. And in any study, the independent variable is the one that the experimenter is going to manipulate, or the experimenter is going to change. And the dependent variable is the one that is going to be the one that's measured to get a result. So in this setting, you can start to imagine how I might create an experiment. Maybe I set up two groups of students, okay? And I have maybe group number 1, and then I have group number 2. And let's say that group number 1, before exam number 1, I tell them to drink maybe 5 cans of 5-hour energy. I would never ask you to do this, keep in mind. And let's say that group 2, I don't want to drink any 5-hour energy. Okay, so the independent variable as we mentioned before, is the consumption of 5-hour energy. And you can see that I'm changing that in this, what is beginning to be an experimental design. I'm having two groups. One drinks the energy drink, the other does not. So I'm actually changing the independent variable in these two conditions of the consumption of energy drinks. And then the dependent variable is the one that I'll measure. So after these students take their exam, I will grade their tests and report the average grade. After these students take their tests, I will grade them and report the results. So the dependent variable is the test scores, and that is the one that I can measure, okay, the average test score of my students. So those are key important things to keep in mind. If you're making a study, what is it that you're going to be changing and what is it that you're going to be measuring? And the answer is independent variable is the one you change, dependent variable is the one you measure. Okay, a really important aspect to a good experiment is to have what we call controlled variables. In the ideal scientific experiment, everything between the variations of the independent variable will be exactly the same. Okay, so what I mean by that is group 1 and group 2, apart from the change in the independent variable, which is the consumption of 5-hour energy, everything else should be exactly the same. So like group 1 and group 2, here's a couple examples of controlled variables. The group 1 and group 2, they should all have the same test administered. If I give group 1 a super difficult exam and I give group 2 a really easy exam, then the difference that I see in test scores might not be due to the consumption of the energy drink, it's probably due to the nature of the exam. Also, things like the time of day that the exam is offered, or maybe you could sum it up as the testing conditions. If I give an exam really early in the morning to one group and really late at night to another group, it could be that the test score difference that I see is due to, just how the time of day affects people's test taking ability. Another thing would be maybe like the demographic of my students. If I give my test to a bunch of students who are really into science, and then I give one group of the test to a group of students who hate science, then you might suggest that you might infer that the test score difference is due to the demographic of students, or even on the same rate, same vein, you'd want to have the same amount of male and female. You'd want to have the same mix of ages and ethnic backgrounds and socioeconomic backgrounds. You want to make sure that your student demographics in the two different trials, the two different conditions of the independent variable, group 1 and group 2, would be the same, right? And we could keep going on. And this is, again, one of the biggest and most important things about creating a good experiment, because you can see if you don't consider the controlled variables, it's very likely that any trends that you find in your data could be not due to the manipulation of the independent variable, but because of the flaw in your research design, your experimental design. So very important things to keep in mind when designing an experiment. And then lastly, oftentimes an experiment will have what's known as a control. And don't let this confuse you. A control, quote unquote, is different than controlled variables. Controls are things that you put in a study. It's kind of like a quality control measure, something that you put into the study and make sure that the components of your study are working properly. So you could have a positive control and a negative control. And the difference between these is that a positive control, the way you could think of it is that it's a situation in which you should get your predicted result, from your prediction here. And the negative control is just the opposite, that you should not get your predicted result. Okay, here's an example. Let's say I am a microbiologist and I am looking to study the potency of some antibiotic. So let's say I take two tubes and I grow up some bacteria in each of these. All right, so here's a tube that's got a broth full of bacteria that I'm growing in this tube, and a broth that has bacteria that I'm growing in this tube. And then let's say I have some sort of antibiotic that I'm testing, so I put the antibiotic in here. Now let's just pause for a minute, and let's imagine that I didn't actually create this tube. I just have one tube that I put the antibiotic in. Well, it could be that when I come back the next day, I find that there's no growth in here, and that would be good because it would probably tell me that my antibiotic perhaps worked. But what would be the case if I came in and I put the antibiotic in the tube and I realized that it didn't do anything? Well, it could be that either the antibiotic doesn't work, or maybe my antibiotic is really old and expired, and it would have worked had it been a good product, but for the sake, but it didn't because of, it was old. So it's always important to include a control. So an example would be like, let's say I grow up a tube that has no antibiotic in it. And then this would be important to know because even in like another way of thinking about it too is, let's say I put an antibiotic in one tube and then I put no antibiotic in the other tube, and then both of them, when I come back the next day, have no growth. All of the bacteria have died in this tube and all of the bacteria have died in this tube. Well, that would also be an important result, because you would know that the reason why the bacteria are dying is not because of the antibiotic, but because of some other unknown feature. Maybe the glassware is contaminated, and so that's killing the bacteria, or maybe the way that they're stored is killing the bacteria, or maybe the way that they're in room conditions are killing the bacteria. It's not necessarily the antibiotic. So including this would be a negative control, because you would not expect there to be any inhibition of bacterial growth if you don't put an antibiotic in there. And the example of a positive control here is a little bit hard to kind of conceptualize. Maybe a positive control would be if you had another tube where you put in an antibiotic that has absolutely been proven and tested before that will inhibit the growth of bacteria. So I could say maybe this has been a proven antibiotic. It will work with certainty. So this would be something like a positive control here. You're going to get your result that the bacteria will be killed by the antibiotic. And then you've got the negative control over here, where you know that the bacteria will not be controlled by the antibiotic because you didn't add any. And then you've got the test tube in the middle. So again, controls just help you ensure that the finding that you observe is trustworthy. Okay. All right. After doing the experiment, then you analyze the results, which is always kind of the fun part. Take a look at the data and see if it supports your hypothesis or not. So the data might support your hypothesis, or the data may not. Okay, and this is a little bit of a tricky thing in science too. Oftentimes, after you've spent this whole time researching your hypothesis, designing your experiment, spending hours and hours and hours doing the experiment, and then collecting all of the data and then analyzing it, there's a temptation in every researcher to try and find that their idea was right. And so this is sometimes called research bias, because you're tempted to look at the data and try and skew it in the lens such that it will help you prove that you were right. But oftentimes you are wrong. And this is one of the great adventures of science, is that you just let the data speak for itself and you follow the conclusions that the data reveal, even if they are kind of confounding, even if they sometimes don't make sense. And generally, most of the famous scientific experiments or discoveries are due because people were looking for one thing, but they accidentally found something else. So you really have to be very objective as a researcher and know when your hypothesis is supported or not. But in either case, each of these are valuable things because they help us understand, help us push forth our knowledge of the natural world around us. So then it's important that you would report your results, let the scientific community know what you did and what you found, so that anybody that's interested in that topic can learn from what you've done. Now, if your hypothesis is not supported, one of the cool things about the scientific method is that it doesn't just end there. You're able to go back and try it again, right? So you go back and you modify your hypothesis to something else. So let's say in the example of the energy drink that I do my experiment and I find that students who consume the energy drink actually did worse than students who did not drink the energy drink at all. Well, then I might have to think more about why it is my test scores have on average been improving, and perhaps it's because of some new study tool that I implemented in my class, or perhaps it's because of a change in the student demographic or what have you. So it could be a variety of different alternative hypotheses to then pursue, and then I would just change my hypothesis and start the whole process again. Make a prediction, design an experiment, analyze the results. And this process just repeats itself and repeats itself and repeats itself. And it's a very powerful tool to drill into what we know about the natural world. Okay, so here's what I would like you to do. Spend a little time practicing this. I've got a couple of scenarios here for you. And for each of these scenarios, I want you to see if you can determine the independent variable and the dependent variable. And then also see if you can come up with some controlled variables, and then think of if any controls that have been included in the study. And here are your scenarios. The first one is that in the 1600s, people believed this theory of spontaneous generation, that like things like mushrooms and grass and animals, they just kind of spontaneously appeared. And according to this thinking, that the reason why flies and maggots would be found on rotten food is because something about the food made them spontaneously appear from the food. And there's a famous scientist, Francesco Redi, who didn't buy this, and he set out to conduct an experiment. And what he did is he had 3 jars, and in one of the jars, in all of the jars, they had meat in the bottom of the jar. But in the first jar, there was no cover so that air and flies could get in. The second jar was sealed so that no air or flies could get in. And then in the third jar, there was covered by gauze, so some air could get in, but not the flies. And then when he examined his results, it was the unsealed jar, the one that was completely exposed to air, that had more flies and maggots. Experiment number 2, this goes back to Alexander Fleming's famous discovery of penicillin. So he was actually a scientist studying the growth of bacteria, and he actually went on vacation, came back and noticed that on one of his dishes that he was growing bacteria on, there was some mold growing. And what was more surprising is that around that area of mold, there was no bacteria growing. Right? So he's growing it on what's called a petri dish, which is just kind of a plastic dish that looks like this. And he's got a petri dish, and there's a spot of mold on there, and there's an area around that spot of mold where there's no growth of bacteria. So everywhere else there was growth of bacteria covering the plate, but then right next to this spot of mold, there's this kind of region right here where there's no growth of bacteria. Well, at any rate, he went and then isolated that and then treated it with a couple of different dishes. On one dish he put bacteria only. On another dish he put bacteria plus the mold, and then on the third dish he just only put mold, not bacteria. Okay, and then these were the results. This is, I think, a hypothetical experiment. I don't think he actually did it just like this, but it's good for us to think about in order to practice this. So he found that Petri dish number 1 had bacteria all over the whole plate. Petri dish number 2, there was bacteria only on the outer edges of the plate, but not near the spot of mold. And then in Petri dish 3, there was no growth of bacteria anywhere on the plate. So consider these. See if for each one you can figure out the independent and dependent variable. Then think about some of the controlled variables that are important. And then lastly, if they have a positive or a negative control. Go ahead and pause it and work through these. Okay, let's see how you did. For the independent variable and the dependent variable of experiment 1, the independent variable would be the thing that you're changing. So this would be something similar to, there's a couple different ways you could word it, but probably something like the exposure to air. The exposure of the jar to air. The dependent variable is going to be the growth of the maggots. And so you can see that what Francesco Redi is doing is changing each jar that has meat in it, its exposure to air. So one is completely exposed, one is completely not exposed, and then one is kind of semi-exposed. So that's what he's changing. So that's the independent variable, again, the one that you manipulate or change. And then the thing that he's measuring, the dependent variable, is the growth of maggots. Now, I'm wondering, what about the controlled variables? Well, you would want to have things like the quality and the type of the meat, right? If you put rotten meat in one of the jars and sterile meat in the other, could be that you get a difference in growth of maggots due to the quality of the meat that you started with. Also, the kind of meat, maybe some meats are more supportive of maggots than others, so you'd want to have the exact same type of meat, maybe chicken or beef or what have you. The same type of jar, and if one jar is full of lime and the other one is not, or if they have different sizes, the size, the shape, the quality of the glass, all of those things could affect how the sterile conditions and could affect the results. And that goes along with the prep. If one jar was prepped by somebody who didn't wash their hands and just threw some stuff into the jar, and the other jar was prepped by somebody that was very thorough, it could be that the growth of maggots that are observed is due not just to the exposure of the air, but to the prep of how each jar was made. And then another thing would be like the location and the environment in which the jars are placed. If one is set next to a cold stream and the next is right out in the baking sun, it could be that the difference you see is due to those. So a whole bunch of different controlled variables. And what about controls? Ones that you would expect to get predicted result and not. Well, Francesco Redi's prediction is that if he increases the exposure to air, it's going to increase the growth of maggots. So positive control is that would be the one with no cover. He's expecting that there should be absolutely a positive result here for his prediction that exposure to air will lead to growth of maggots. And then the sealed one is kind of like the negative control. He would not expect there to be any growth of maggots observed when there's no exposure to air. Okay. All right. Experiment number 2, we've got the independent variable, the dependent variable. Independent variable would be a couple different ways you could word it, but probably the best would be the let's see here. The dependent variable would be the inhibition of bacterial growth. That's what you're measuring. And the independent variable would be the presence of mold. Okay, so he's changing whether or not there's mold added to the plate and looking to see how it inhibits bacterial growth. Controlled variables would be things like you'd want to make sure you have the same type of bacteria. If you put one type of bacteria in one dish and another in another dish, it could be that the lack of growth is not due to the inhibition of, or that is not due to the effects of the mold, but maybe perhaps because one bacteria grows in a certain condition versus another condition. You'd want to have the same type of mold. If you just grab mold off of a piece of bread and throw it in a petri dish versus the special type of mold that causes the effect, that would skew your results. You'd want to have the same type of petri dish, the same type of nutrient base, and you kind of get the point there. Again, the only thing you change is the independent variable. You try and keep everything else exactly the same between all of the different conditions of the independent variable. All right, and then lastly, the controls here. You could think that this one is probably a negative control. You would not expect there to be any inhibition of bacterial growth if you don't add any of the mold to it. And then this would be kind of the positive control. You would expect absolutely that there wouldn't be any growth of bacteria if all you have on there is the mold, and the mold is the thing that is inhibiting the bacterial growth. So you should expect to have complete inhibition of bacterial growth if all you have on the plate is the mold, okay? And then these are kind of your quality controls, so then Petri dish number 2 is the thing that really gives you the information that you're looking for. So a little bit of a tricky stuff here, but this is the scientific method, and this is a very powerful method because it keeps bias out of the equation, and it kind of commits all of us to objectivity, which is the thing that really helps discovery of different phenomena in the natural world. All right, one last quick note here about how scientists would report their work. This is called the peer review process. So you may have heard this word if something's peer reviewed. Basically means that a paper is submitted to a review committee at some sort of scholarly journal. Journals, there's a whole bunch of them out there. A couple examples is one in science that's really popular is called Nature. Another one is just called Science. And you generally find that these journals, the ones that have the simpler name to them, are usually the older ones. So these ones have been around probably for hundreds of years. And then sometimes you even get like more new ones that are really specific. Like this one says the International Journal of Neuropsychopharmacology. So that's a very specific branch of biology. In either case, these are kind of like magazines or just periodicals where there's people that work for the journal, and they receive all of the submitted work from all of the scientists out there submitting work to them, and then they are going to review them. Okay, so on these review committees, let's say I wanted to report my findings, so I submit it to the journal Nature, and at the journal Nature, they've got a committee of professionals that are experienced, and they're going to look at my paper, and they're going to make sure that my research design is good, that my data was taken in an accurate manner, and that the interpretations that I'm getting from that data are valid. And if it's not, then they will provide me with some feedback, and then I will have to revise my paper to suit the likings of that committee's feedback, and then I'll go ahead and submit it again to them, and then it may or may not meet their expectations, and they might send it back to me, and then I'll have to do it again and again and again, and then eventually, if my paper gets to the quality that satisfies all of the people on the review committee, then they will accept it into their journal, and it will be published for all of the scientists out there to read. Now, the way that these journals make their money is that they charge a subscription to view all of their content. So like that journal Nature, the journal Science, they would require that you pay a monthly subscription to read their work. But lucky for us, there are these things called databases, which are going to give us access to all of these different types of papers. And here at the library at St. Paul College, it's likely we have a subscription to one of these databases where we can actually access all of those published works. One that's free is Google Scholar. You just type in Scholar and Google the word Scholar, and Google Scholar will pop up. And then it doesn't give you access to all of the publications out there, but it does give you access to quite a few. In either case, this is kind of the nature of professional scientific work. All right, now we're going to proceed from kind of this overview of the scientific method, and we're going to get on to talking a little bit more about biology-specific things. First and foremost are the properties of life. There are 8 properties that all living things share. The first is order. What I mean by this is the fact that the way that my cells behave and are arranged is very similar to the way that your cells behave and are arranged. And it's not like one day my cells behave a certain way that's totally contrary to yours, and then the next day they change. There's a lot of order to how things are arranged. You know, all things are made up of atoms and molecules and organelles and cells, and that doesn't change from organism to organism. Another thing is that all organisms are going to have some ability to respond to their external environment, and this can be either really complex or rather simple. For example, a plant, even though it doesn't have a brain or eyes or nose or taste buds, it can still detect where sunlight is coming from and orient itself accordingly. It's always fascinating to me how they do that. This is a picture of olfactory neurons in your nasal cavity. So each one of these little things can detect smells. As a little smell molecule lands on them, it can process it and send that information to your brain. Here's a picture of the rods, these ones here, and the cones that line the back of your retina. So as light strikes your retina, it strikes these little receptors, and then they translate that information as vision. Even some of the most simple organisms have a really cool way of responding to their stimuli. This is a euglena that is a single-celled animal, a tiny, tiny single-celled animal. It's got a little eye spot here, that little red thing that you see that can actually detect greater or lesser concentrations of light. And then it knows how to swim accordingly to get better access to light so that it can photosynthesize that light and make energy. So a lot of variation there in how complex organisms can respond to their environment, but nevertheless, all organisms can respond to their environment. Another attribute that all cells have, or all living things have rather, is their ability to reproduce. And this could mean something like cellular reproduction, such as the process of mitosis and meiosis. We're going to learn more about those later in the semester. Or it could even mean organismal reproduction, so the ability of a sperm and an egg to fertilize and create an embryo and a fetus. All things can adapt. What this idea means is this kind of gets us to the idea coined by Darwin in the 1800s, basically that organisms have a variety of traits, and generally, the variation that we see in the traits out there is the thing that helps organisms survive in one climate versus another climate, or one situation versus another situation. His big discovery that he's most probably famous for were these finches that he found. So he boarded this big ship and went to the Galapagos Islands, where he found a variety of different types of finches. And some finches had a smaller, narrower beak like this one, and that was better suited to eat insects or to get the nectar out of flowers. Some of the finches had a big beak like this one, which was well suited for cracking nuts. And then where these birds scattered was depending on the food source available. So that was kind of one of the big first examples, but there's a whole bunch of examples of adaptation. Like another example would be this organism, which is very well adapted to live in any kind of environment where you have leaves of this shape and size, because this thing looks just like a leaf. And another example would be like in caves where there's no light. Organisms have lost their eyes because their eyes don't do any good in a place where there's no light. And so instead they have extraordinarily well-developed pressure receptors on their skin for detecting minute changes in pressure, and that's how they catch their prey. And so there's all sorts of really cool adaptations out there. I mean, when you look at all the different plants and animals and organisms, there are so many cool kind of features that they have that are really diverse, and they're not shared by all organisms. So a unique feature of an organism that allows it to survive in its environment is what we would call an adaptation of that organism. Okay, all things can grow and develop. That's pretty self-explanatory. All organisms have an ability to regulate. This means if something's too low, they can get more of it. If something's too high, they can get rid of it. An example would be like my blood sugar. If I have a low blood sugar, my body is able to raise the blood sugar, and if I have a high blood sugar, my body is able to lower the blood sugar. So that's one example, and this runs really deep. Cells are really good at regulating themselves. If there's a cell that's trying to survive and it needs a protein to make something to keep it alive, it can use DNA to make that thing. If a cell has too little or has too much of something, it can break that thing down. So cells are turning stuff on and turning stuff off to regulate themselves all of the time. And we're going to learn a little bit more about how genes do this and how DNA is utilized to help a cell regulate everything that it needs to survive. And this idea of regulation goes kind of hand in hand with this fancy word, which is called homeostasis. Homeostasis, the word literally means an unchanged standing. And what it refers to is the tendency for organisms to want to stay at a set point. So an example would be, again, using my body as an example, my body wants to stay at a very narrow set point at about 37 degrees Celsius or 98.6 degrees Fahrenheit. And if I get too cold, I'll start shivering to warm my body temperature. If I get too hot, I'll start sweating to lower my body temperature. Okay, so it's my body wants to stay at a common temperature, and it wants to stay at a common level of salt in my blood, and I want to stay at a similar blood pressure, and I want to keep my hormones at about the same level. So your body and the cell conditions of the cell and of all organisms, it usually doesn't like to change all that much. It usually wants to stay the same, and then the organism will do what it has to remain at that set point, whether it be temperature or chemical balance or what have you. And then lastly, all organisms can make energy or process energy, so it takes energy to keep something alive, and so an organism needs to be able to do that. All right, so let's talk a little bit about how living things are arranged. Kind of the simplest fundamental thing out there is the atom. Okay, this is kind of the most indivisible unit of matter. When I say matter, I just mean all of the stuff that's out there. By the way, we really have no idea what anything is out there that exists. It's kind of all that exists out there is pretty much a complete mystery. But at a certain level of complexity, we have identified this thing, the atom, and we know that matter is made up of atoms. What atoms are made up of, who knows? They're made up of these things called quarks. But then beyond that, some people think that they're made up of these little vibrating strings. That's known as string theory, and who knows what all matter is. It's all kind of a great, wonderful mystery. But after atoms, then atoms make molecules, and molecules are things that we can actually see with given the right instruments. And molecules are basically made up of a bunch of atoms. So if I take a bunch of atoms and link them together, it forms a molecule. And now if I take a whole bunch of atoms and link them up to make a really big molecule, then we call that a macromolecule. And in biology, there's 4 main classes of macromolecules. There's fats, sugars, starches and sugars, proteins, and nucleic acids. We're going to talk more about that later in the class. Just to give you some scope here, this is a little molecule of water. Water is H2O, which means it's made out of 2 atoms of hydrogen and 1 atom of oxygen linked together. Some of these other that I've included, these are macromolecules. This is like a fatty acid, so the fats in your body look like this. There's just a bunch of carbons and hydrogens linked together. This is an example of a protein, a bunch of amino acids linked together. Okay, if you arrange a whole bunch of molecules and macromolecules together, it forms organelles, and organelles are the things that are found in living cells, specifically eukaryotic cells. Prokaryotic cells don't really have organelles, but we'll talk more about that later. But in either case, here's an example of an organelle that you and I would have. This is called mitochondria, or this is a picture of a mitochondrion, and basically how it works is it makes energy. It takes oxygen and makes energy. So the reason why you're breathing oxygen right now is because in the trillions of gazillions of cells in your body, you've got lots of these mitochondria, which are taking that oxygen, making energy out of it. So your mitochondria thank your lungs for taking that oxygen in. Just to give you some context, this is what a picture of a cell, and you can see, these mitochondria are found here and here and here, but there's a whole bunch of other stuff inside of a cell as well. And it's at this point that I like to pause and just point out something. A cell is like a microcosm, all right? The capital is the nucleus, and there's a whole bunch of stuff in a cell. There's a production factory where stuff is made. There's the energy powerhouse. There's the packaging center where stuff is shipped out and processed. There are the places where proteins are made, or these little dots here. We're going to learn more about all this stuff, but I just want to point out how complex a cell is. It's literally like a little city in each living thing to make all the stuff that it needs. And there is more information in the inside of a cell than probably all of the books out there in the world. The amount of information encoded in DNA to make your entire body is incredible. Just think about this for a minute. All of your bones, the shape of all of them, all of the muscles, the shape of all of them, and how they're arranged, and all of the microscopic structures, all of your nerves, all of your blood vessels, all of your organs, and how they all have these extraordinarily complex shapes, and all of the tubes that run in and out of your body. That the instructions for building all of that is all in this tiny, teeny, minuscule little spot called the nucleus of a cell, where there's DNA here. That blows my mind. And that's in one cell. And if you look at your hand, there are trillions of cells, just, probably millions of cells just on the skin surrounding your hand alone. And so the fact that you and I are alive right now is what I feel to be nothing short of a miracle. Okay? But in either case, cells are made up of organelles, if they're eukaryotic. If they're prokaryotic, they don't have organelles. But in either case, all of the stuff inside of a cell is made up of molecules and macromolecules. If you put a bunch of cells together, it forms a tissue. So an example of this would be like this is skin tissue, and all of these cells are called epithelial cells. They're a certain type of cell, and when you have a bunch of similar typed cells together, it forms a tissue. Another example would be this is fat tissue. All of these cells are fat cells, and fat cells all behave in a very similar manner, which is different than skin cells or any other kind of cell. So we call that fat tissue. Here's a picture of muscle. These are what muscle cells look like. And if you add a bunch of muscle cells together, it makes tissue called muscle tissue. And the way that our tissues work depends on how the cells function. Okay, organs then are when you create a couple of tissues and you add them together, it forms an organ. So an example here would be like the heart. The heart is made up of muscle tissue, but it's also made out of the tissue that forms the arteries and veins. There's also some fat tissue on there, so lots of different types of tissue there, making up an organ. But when you add a bunch of organs together, it's called an organ system. So whereas the heart was an organ, the cardiovascular system is an organ system made up of the heart plus a bunch of other stuff, a bunch of other vessels, which are considered organs as well. Okay, you add a bunch of organ systems together, you form the organism, and then you add a bunch of organisms together, it's a population. You add a bunch of different types of populations together, it's a community. Okay, so this is kind of the grand scheme of how life is arranged. Life is also very diverse, and trying to figure out what an organism is and how it fits into the diversity of life is kind of a tricky business. But so far our working model is what's called a three-domain system. So you take any living thing out there, whether it be a bacteria or a fungus, a plant, a protist, animal, whatever, a fish, you can divide any living thing into one of these three domains. There's the eukarya, which are the eukaryotes, meaning that all of their cells have organelles. There's the bacteria, which you probably know what a bacteria is. And then there's the archaea. Now, archaea and bacteria are pretty similar to one another, and they're both microscopic, single-celled organisms. Whereas the eukarya are the things that are a little bit more, you can see them. These are things like fungus, animals, plants, and so forth. Inside each of these, there's even a further way we can classify organisms. And this is known as hierarchical taxonomy. And it was an example of this would be, let's say we have a fox. Okay, this particular fox drawn in the picture is known as the red fox. And there's a bunch of different other types of foxes that are not red fox. Well, every organism has what's called a binomial name. And the binomial name is in Latin, and it looks like this right here. So the common name would be red fox, but the binomial name would be this Latin word vulpes, vulpes. Okay. The first word of a binomial name refers to the organism's genus, and it's usually capitalized. The second name in the binomial name refers to the species of the organism. And every organism has one of these names. Like for us, we as humans, we have the binomial name Homo sapiens. Sorry, and Homo would be our genus, and sapiens would be our species. Now, just to give you some context here, this particular organism here, the red fox, it's Vulpes vulpes. So Vulpes is its genus, and coincidentally, Vulpes is also its species. But this organism is in the family of the canines. The canines are in the order of the carnivores, meaning the meat eaters. The carnivores are a subdivision of a bigger class called the mammals. And the mammals are part of a phylum known as the Chordata, those things that have a notochord is what it's called. These are all animals, so they're part of the kingdom Animalia, and then that's part of that, the three domains, the eukarya, because the cells are eukaryotic. So sometimes the thing that I remember learning in middle school was kings play chess on funny green squares. If you can remember that mnemonic, kings play chess on funny green squares, you can remember these different classifications and the order of specificity of each. And the big thing to keep in mind is that, like a kingdom, or I'll put it this way, all things in the kingdom Animalia, all things in the animal kingdom are eukaryotes, but not all eukaryotes are animals. Okay, so you have to know the order of specificity. Another example would be all organisms that are carnivores, meat-eating animals, are in the class of the mammals, but not all mammals are carnivores, meat eaters. Okay, or you could say not all canines are in the order carnivores, but not all carnivores are canines. So just know this level of specificity. This whole scheme was first put forth by this guy named Carl Linnaeus in the 1700s, and it was a great tool to be able to kind of make sense of all of the diversity that we see out there. And this is kind of an area of science that's changing a lot, specifically in things like single-celled organisms or plants and animals and things like that. In fact, this three-domain system here, this was recently just put forth in the late 70s. And prior to that, there was just what was known as the five-kingdom system. And so it is changing. As we learn more about organisms, we figure out better places to classify them. And that is all that I have for you for Chapter 1.